So thank you all for joining us today for seminar. It's my great pleasure to introduce Dr. Colleen Burge, who was my external committee member. Dr. Burge received her BS and PhD in aquatic fisheries sciences at the University of Washington. Her dissertation focused on host pathogen environment interactions of oyster herpesvirus and the Pacific oyster. After she completed 2 postdoctoral positions, first at Cornell University in the Department of Ecology and Evolutionary Biology under Dr. Drew Harvell, which connected me with Dr. Burge for her work on pathogenic Labyrinthulomycetes. Then Dr. Burge held another postdoctoral position at the University of Washington. So she's now assistant professor at the Institute of Marine and Environmental Technology with dual appointments at the University of Maryland, Baltimore County in the Department of Marine Biotechnology and the University of Maryland, Baltimore in the Department of Immunology and Microbiology. Her lab of aquatic animal health focuses on marine host pathogen environment interactions, including disease ecology, organismal physiology and immunology, and the development of disease diagnostics. Dr. Burge has worked on a variety of diseases affecting marine organisms, including the Pacific oyster, manila clam, sea fans, corals, sea stars, eelgrass, and seagrasses. So today, Dr. Burge will tell us about 2 host pathogen interactions in a changing ocean. So I'll give it over to Dr. Burge.
Thank you, Sabrina. Thank you for the wonderful introduction, including my multiple affiliations. So today, what I've done, I have so much data from both of these systems, but I think it's nice to spin it in a positive way. So I'm focusing more on some of our applied research. So we've also done a lot of basic research in both of these systems. Let's see here. Of course, now it doesn't want to advance. So as Sabrina mentioned, my lab focuses on host pathogen interactions in a changing environment. And we focus on 3 major themes within this disease triad or triangle. I really like the triangle because it depicts change. So factors in the environment such that drive disease and also climate change impacts. Factors in the pathogen zone such as diagnostics and spread. And also in the host realm such as biomarkers for resistance and immune response. And we've worked across multiple different systems, so working on oyster pathogens, eelgrass pathogens, as well as the interaction of oysters and eelgrass. And the second half of my talk, I'll talk about potential for eelgrass to mitigating oysters to mitigate an eelgrass pathogen, and we've also done some work on corals. So I think I always show this side of the major systems of study that I've worked on, and I think especially for students, it's really important to understand that going outside of your comfort zone is really important. So as Sabrina mentioned, I worked on Pacific oysters and the oyster herpesvirus for my PhD. That is a system that I'm again working on in somewhat different capacity. When I went to Cornell, I originally went to work on sea fan corals and what turned out to be a pathogenic Sonoplanochytrium and also a fungus Aspergillus sodowii, which actually led into a similar pathogen Labyrinthula zostra in Zostra marina. And I also worked at the tail end of my postdoc at Cornell on mass mortalities of sea stars. But I can tell you that this breadth of study really helped me. So students, when you're starting to think about moving on to the next step, I think that's really important. But for today, I will touch on these 2 systems. So Pacific oysters and the oyster herpesvirus first, and then Zostra marina and Labyrinthula zostra. So what is OsHV-1 and why should you care? So the Ostreid herpesvirus 1 or OsHV-1 causes mass mortalities of Pacific oysters. And in fact, the host range is not restricted to Pacific oysters. And I'll discuss a little bit more about that later in the talk. The disease progresses quite rapidly and can kill up to 100% of animals dependent on the family or stock. So there's a genetic component to this disease. Once established, OsHV-1 persists in the environment, causing seasonal mass mortality. So this is one of those cases where we see mass mortalities at the height of the summer months when temperatures are really warm, typically. The virus can be transmitted within species and life stages, both horizontally and vertically. And there are multiple variants of OsHV-1, including what are known as the OsHV-1 microvars, which are emerging globally. So everyone literally that I ever talked to asked me if they can get OsHV-1 from eating a raw oyster. And I'm sorry if you've heard this joke from me before, but I really hope that you're looking at this evolutionary tree, which includes the 3 branches of the order Herpesvirales, and see that the molluscan herpesviruses, which include OsHV-1, as well as the abalone herpesvirus, branched off early evolutionarily. And in fact, only share 1 gene in common. And this is not a gene that would have anything to do with infection processes. So they split early from the terrestrial vertebrate viruses that include our human herpesviruses and the other fish and amphibian viruses. So why are they grouped together? Well, they are morphologically similar. So OsHV-1 and this down here is herpes simplex virus 1 are morphologically similar. We are very fortunate this tree is possible because we have a full genome for the reference strain of OsHV-1. Do not know why advancing slides is, oh, there we go. So if we step back a little bit and go back into the early 90s, the herpes-like viruses were described in mortalities of larval oysters in Pacific oysters in France and also in New Zealand, followed by mortalities in juvenile oysters, and that sequencing, that full sequencing of the genome that really led to development of molecular tools to work on this virus. This helped us to first detect it in Tomales Bay, California in the early 2000s, and we believe it was linked with mortalities as early as the early 90s when mortalities were first detected. Followed by detection in multiple Asian countries by my colleagues at BIMS, though there was less known about the impacts of OsHV-1. And you'll notice there are no stars on the east coast of the US because to date, the virus has not been detected on the East Coast, which I think is particularly important, though there have been few surveys to date. There was a survey in the early 2000s, so about 20 years ago. So what are the OsHV-1 microvars? In 2008, severe economically devastating mortalities were reported first in France caused by what they named OsHV-1 microvar. And what was different here, instead of just affecting larval and juvenile oysters, these microvars can kill oysters of all life stages. Now we understand this is not simply one microvar, but many microvariants. And probably in the context of COVID, you understand there are multiple variants of SARS-CoV-2. So this probably makes sense to you. And I'm not going to get into the specifics, but these are specific genotypes that show variation in genome regions compared to this OsHV-1 reference. And if you've heard of Palm's disease in Australia, this is a causative agent of Pacific oyster mortality syndrome. And I think I've already mentioned this, but there's really a high level of concern about spread of the OsHV-1 microvars. And I'm seeing increased concern in the US as time passes. And why is this? So after this first detection in 2008 in France, the virus was moved actually throughout Europe a lot through animal transfers. They see mass mortalities yearly, for example, in Ireland. But even more alarming was the detection in mass mortalities in the North Island in New Zealand, as well as Australia in 2010 with no known transfers. And the data to date indicate that these are slightly different microvariants than the original, and followed by movement of this virus, again with no known transfers in 2016 to Tasmania and 2018 to South Australia. Similar microvariants have been detected in Asia, and again, we don't have a good idea of how these microvariants fit within the grand scheme, and not a lot of work has been done there. A lot of the work is being done in both Europe and Australia. And later in the talk, I'll talk about the first detection of an OsHV-1 microvariant in San Diego, California, which has been some of the focus of my lab over the last couple of years. It's been an interesting time of learning about management policies and next steps, et cetera. So in fact, how do you manage OSHV-1? Well, it's really not practical to cull the oysters, and we don't honestly know what the vector or the ultimate host that leads to transmission yearly. And because oysters lack an adaptive immune system and only have an innate immune system, traditional vaccination is not possible. So they lack the B cells and the memories, so the memory cells. So, and you might also imagine it would be really difficult to vaccinate all of those little tiny oysters as well. Recent work has indicated a virus mimic does enhance survival, but the long-term health effects are not well understood. You can restrict movement of animals. I think this is extremely important and to better manage health of animals. You can educate end users on risks if OSHV-1 is detected and improve the speed of detection, which can be quite rapid because this is a qPCR assay. Something that's worked really well, particularly in France and Australia, is breeding for resistance. So there is a genetic component of resistance. And in fact, in France, they have families that can clear and eliminate the virus. And also determine genes involved in OSHV-1 resistance to more broadly be able to understand and be able to breed for, especially for resistance, especially in areas where the virus is not endemic. Can determine species more resistant to OSHV-1. So this is primarily a Pacific oyster disease, and Pacific oysters are the most cultured species in the world and make up much of, for example, make up much of the West Coast culture systems, but there are also multiple other species that could be used. And also in closed systems, water treatment can be used to limit infection. So in the United States, I've already hinted to this. So the primary two species are the Pacific and the Eastern oysters. And probably most of you are quite familiar with the Eastern oysters on the US East and Gulf coasts and many of their pathogens and issues. In fact, the Eastern oyster is also grown on a small scale on the US West Coast. But on the US West Coast, the Pacific oyster was introduced and is the primary species cultured. However, the smaller Kumamoto, the European flat, as well as the native Olympia oyster to some degree are cultured. So there are some different species that can be grown. So our research objectives for this part of my talk were primarily to conduct laboratory trials to examine differential survival of oysters to OSHV-1 microvirus and OSHV-1 Tomales Bay, using multiple stocks and species, and also an initial characterization of a new OSHV-1 from San Diego, California. And this work was primarily a NOAA Sea Grant project, as well as some funding from the USDA ARS. And just so you understand, when I say OSHV-1 TB, this is OSHV-1 Tomales Bay. This and nearby locations of Drake Sestero and Bodega Bay until very recently were the only places where OSHV-1 had been detected. And so this OSHV-1 TB just stands for this non-microvariant. I've been working in this system for nearly 2 decades now, which seems crazy to me, but is actually the truth. So as I mentioned, we did some laboratory trials to look at the potential resistance of existing stocks that were used in the US. And first of all, we had done a field experiment, which I don't have time to talk about very much. And we used many different stocks and species, but for this laboratory trial, we picked 3 stocks of Crass ostriogygus, so CG1, 2, and 3, which are broadly used by the US West Coast industry, as well as some stocks of C. virginica, which we couldn't actually plant in Tomales Bay, which are probably familiar. So the Debbie high salinity and the low salinity that are widely used by industry. So this graph depicts our survival in the field. So the CG1, 2, 3, and Crassostreicomia, and their mean survival. And we often use multiple outplants because we see disease pressure at different times. So we saw with CG1, I was hopeful that because it had high survival in Tomales Bay, or as compared to other stocks, that it would have high survival against the microvariants. CG2, I thought would have low survival. It had low survival in Tomales Bay overall. CG3, moderate. And I was very hopeful for Crassostreicomia to have high survival, as well as the 2 Crassostrea virginica stocks. And just so you know, I always have to give this little slide that while we're really worried about the microvariants because they're more virulent and they're able to spread more, this bag number 88, there had been 500 oysters in this bag just 2 weeks previously. And you can see this small pile here. And by the end of the summer, there were about 10 oysters. So don't discount just because it's not a microvariant that it's not an issue. So we ran our laboratory trials with the OsHV-1 microvariants in Tucson, Arizona. And just to give you a sense, we had to have permission from the state veterinarian as well as the USD APHIS. So we imported virus stocks from the highly virulent virus stocks from France and also from Australia. And the reason for running these trials in Tucson, Arizona was not because I love saguaro cactus or because I love hiking, which are both true, but in fact, we wanted to make sure that there was perception that we were not bringing in these variants and we were not having it, making any issues for the oyster industry. And there's actually a famous shrimp lab there that we've now been working with for several years. In this injection trial, so again, we have our 3 stocks of C. gidus in the same colors, the C. sycamia and the 2 C. virginica stocks. And I will show a few slides like this with day on the x-axis and surviving on the y-axis. So these are Kaplan-Meier survivorship curves. These are animals injected with the Australian microvar. These are the French microvar. The control is not shown, but they had 100% survival. And first of all, if we look at the C. gidus, we see that most of them did not perform well. So not even the one that I was very hopeful for. Interestingly, one of them that I thought would be moderate had flipped survival, whether the Aussie or the French microvar. And interestingly, also Crassostreicomia. So they had moderate survival to the Australian microvar, and relatively high to the French microvar. And colleagues in France had also done some trials with Crassostreicomia and seen similar results. Now, what you all are probably interested in are the Crassostrea virginica. And what we found was that so CV2 had high survival, no losses, and CV1 had about 10% losses to both microvars. We followed this up with using quantitative PCR as a proxy for infection. This is a non-culturable virus, so this is our best tool to understand relative copy numbers. And what we saw, just to walk you through this, first of all, these are all of the animals that died with injection with the Australian variant. And this axis is 0 to, I believe it's 100 million. So most of them had between 1 million and 10 million copies per milligram of tissue. This is a lot of virus. This is exactly what we'd expect if these animals were dying of OSHV1. And you'll notice that all of the stocks, including that CV1, which had 10% mortality, had high copy number. If I, let's see here. And the survivors generally had much lower copy numbers. Though some of the survivors did have much higher copy number, even in species, in the C. sycamia or the virginica. And we didn't see any differences between stocks or species, so this was a relatively small sample size, but there was 1,000 times more OSHV1 DNA in mortalities, as is pretty apparent based on this graph. So we've also used what kind of a more natural exposure technique. So in the previous trial, I showed you data of oysters that were injected. So it bypasses their primary defense system. And that was because those oysters were about this big and they would be very difficult to work with in the lab and very difficult to have enough virus to work with. But there is another method that we've been using more recently. And this is, as I said, a natural exposure technique. So in this case, once again, Tori is showing us injecting juvenile oysters, and we inject them with 1 million copies. We put them in tanks. We let the virus replicate for 24 hours. And we netted this much virus per mil, I believe, from the French microvar, the Australian, and then that Tomales Bay non-microvar. So we were comparing 3 different microvars. And then we simply added water from these tanks, which had antibiotics, into plates of these nice, small, teeny tiny oysters, which are about the size of my pinky nail. And we exposed 20 families that had been previously determined to be either high or low surviving based on an experiment in France. So these are molluscan broodstock program animals. So this is a genetic breeding program on the US West Coast. And we looked at, of course, we looked at survivorship as well as qPCR for tissue and water. I believe I only included the tissue qPCR here. What we found was somewhat expected. First of all, other than this one family, which turns out to be not the best family, I think it was excluded from further analysis, that the controls had high survival through 6 days. Those exposed to OsHV-1 Tomales Bay were not significantly different than the controls. And this was not unexpected. Prior to the OsHV-1 microvars, no one had been able to complete transmission to small seed or juvenile oysters in the lab. However, the interesting thing was we were able to kill oysters with injection. So I think with a little more work, we might get somewhere with this. But I think the really interesting component here is the mortalities to the Aussie and the French microvars. First, they were significantly greater than the controls in Tomales Bay, and I think it's pretty clear that there was a lot higher mortalities to the French microvar. And I just wanted to point out there was high tissue copy number again in that kind of same range among morts to all of these viruses. And if you are part of a breeding program, your interest would be here to pick high survivors. And we did have some high survivors to both the French and Australian microvars, but it's not a perfect correlation. There are some families that do better or worse against different microvars. And of course, acknowledging we know that we expose them to different amounts of the virus. So, you know, that's kind of mimicking a natural transmission. In future studies after this one, we actually did our best to equalize the virus concentrations. So what about our qPCR data? So one of the low surviving families was removed from this analysis. Remember the one I showed in the controls that didn't do very well. And I've squared out the ones that are expected to be low survivor. What we found was the French and the Australian microvars were statistically different from each other. And again, there were greater copy numbers than the Tomales Bay or the controls as we expected. However, interestingly, there was no differences among families or days. And these are living oysters that were collected on day 4 or 6 of the trial. And we found this interesting because these could be survivors. We don't know. It's not the end of their life, but they have a lot of copies of the virus in their tissues, which indicates to us that these animals are tolerant rather than being resistant. So, and this is really not expected. The true resistance to something like OsHV-1 would, from a pathogen standpoint, would take several rounds of selection, which we're really not, at this point, this is a first step. So if we go back to detection in the US, again, I'm focusing on the US West Coast instead of the East Coast. Again, we don't believe that there are OSHV-1 issues on the East Coast. And this is my star for Tomales Bay with the non-microvar. And I think it's really important to understand that this industry is interconnected and really works based on their high health status and being able to move animals between locations. And that not only includes what you might think of Washington, Oregon, and California, but also Alaska and Hawaii. Of course, no animals are moved from Tomales Bay. And I think this is one of the reasons why the industry is, to date, for the most part, been OSHV-1 free. The more recently, in late 2018, I'll talk about the detection of an OSHV-1 microvar microvariant in San Diego, California. So this was a new aquaculture venture looking to establish health history, and they were using a collaborator, Ralph Elston at Aquatechnics, to test their animals, and they had been putting animals out between early 2017 through 2018. In fact, they were just about at their 2-year history to be able to start selling animals and move animals at least within Southern California. In October of 2018, I received a phone call from Ralph Elston that they had detected OSHV-1 in a batch of Pacific oyster triplets in all of the pools of animals. And so this is how I became involved, and I was asked to look at what the virus variant was, and which I'll tell you a little bit more about. And following that, in December, again, the virus was still present. And at this point, all of the animals were removed from this aquaculture venture. So it was a nursery system. So this was the first detection of OSHV-1 outside of Tomales Bay area in the US. And again, this was a new aquaculture venture. And so following this, we aimed to characterize the variant and conduct a pilot transmission study to determine whether the newly identified variant was infectious. And I think this is often extremely difficult and also hasn't been done as much as it probably should be done with different OSHV-1 variants. So after Ralph's lab sent me the samples, we used a specific genome region again that can delineate OSHV-1 from the microvariants. And I sequenced everything I had in the lab from Tomales Bay, as well as the variants that we've been working on from France and Australia, just to make sure that there was no contamination. And also this tree is also based on, I picked some in GenBank that were a wide geographic spread. And what we found were that, as expected, Tomales Bay, Mexico, grouped more closely with the reference strain, while all of the microvars, including OSHV-1 microvar San Diego, which is what I named it, grouped in the lower part of the tree. So this, along with my initial sequence analysis and alignments, really indicated that this belonged within this group of viruses, though we don't honestly know that this is the best genome region to be looking at. So what happened following detection? So the FLEPSI was depopulated and shut down. And the FLEPSI reported 99% mortality in that group after that October sampling. And honestly, the copy numbers were extremely high. And you'll notice this is in the range of what I showed for experiments. The original seed source was tested in the heat test of juveniles coming from the source bay, which was in Northern California. And there have been ongoing sentinel studies both in San Diego and other locations on the US West Coast. So we were able to show the infectious nature of this virus. And first on the left is another one of these Kaplan-Meier survivorship curves. And I made 0.2 micron tissue homogeneous, just like I had previously for the French and Australian microvariants. And we were able to get some groups of oysters from one of our farm collaborators, some diploid and triploid stocks. And they were either injected or exposed. So there were different tanks for injected and exposed. And just to note, this was really a pilot, and the copy numbers used in the experiment were really low. I honestly never expected it to work, but lo and behold, it did. So if you're looking at this curve, you see that especially in blue, this triploid had high mortalities in both the injected and the bath. And when we move over and look at the qPCR copy numbers in the moribund animals, they were extremely high. I have since taken tissues from this experiment and created new homogen and exposed additional oysters and done additional trials. So for the next subsequent, I don't know, year and a half, OsHV-1 was not detected in San Diego. So they did various sizes and species soaked at the FLEPC, but then during a sentinel study, so there's been a West Coast sentinel study as an early warning system looking at locations in Washington, Oregon, and California. Again, we detected OsHV-1, and in fact, all of the animals pretty much died in these two families of oysters. And just to give you a sense from this sentinel study, and this was conducted in summer of 2020, this is one of the first things we were allowed to do in the lab. Just quickly to walk you through this, this is two different lines of animals. Again, this is OsHV-1 copies per milligram of tissue. This line depicts a threshold for what's expected for mass mortalities in France. Anything with a red bar is something where we had fresh dead animals as shown in this picture. And anything with these non-NS in the skull and crosscone means there were no animals left. So this had started out with a few hundred animals or a couple hundred animals per basket. Within 2 weeks, there were this line B had very few left. And then by, I guess it was about a month after deployment, we literally received the last few animals and couldn't even fill in all of the samples we were going to collect. So that gives you an idea of how quickly this can happen and how rapidly the virus can replicate. And our sequencing, as well as sequencing from the US National Reference Lab, showed it was identical to the 2018 detection. So what are our next steps in working on this system? Though data indicate non-Pacific oyster species are less susceptible, we really haven't looked at the very susceptible life stages, so the little guys. I have done some additional trials, but I think more work needs to be done. And in fact, I just received funding from the USDA aquaculture to look at C. virginica, specifically to both look at susceptibility of families, as well as to look at whether there's a genetic basis for resistance. There needs to be additional characterization of OsHV-1 from San Diego. It's currently being sequenced, so we can look at it comparatively, both with the Tomales Bay as well as other sequence microvariants. It would be nice to have a high throughput assay rather than to clone and sequence or sequence PCR products every time. We're also working on a gene expression analysis of Eastern versus Pacific oysters from our original experiment, the injection trial. So what do we really need to do in the long term? I think we need to pair laboratory and field trials with quantitative genetic studies to really identify those markers of resistance and susceptibility. And this is really a lot, it has to do with the fact that for the most part, our culture systems are outside of the endemic zone. So to be really ready for these viruses, we have to have tools developed. And of course, our end goal is to reduce potential for introduction. So what are the future effects of OsHV-1? There is a lot of concern, or there's a lot of global strain variability, which we don't understand very well. There's a rapid emergence in mortalities in OsHV-1 microvars in some locations. There are environmental factors associated with these mass mortalities, though it can be different dependent on variant or ocean basin. Again, this is really something that happens in the summer months. This news story somehow always pops up and is sent to me, and it kind of makes me upset because we don't have enough data to say it's the latest symptom of global warming, but I think it's the latest symptom of globalization, maybe, is a better terminology here. And of course, I highlighted, management strategies already. I mean, there are other types of management strategies that can be used. I've been happy and with so far what's been done in the US, though I think a lot more testing needs to be done. And I know that on the West Coast, the regulators are really cognitive, as well as the shellfish farmers, because they don't want this virus spread. And I get the general sense from the East Coast farmers as well. So I am well aware that climate change can influence marine diseases. I've written reviews on this, so that last point was kind of a ha-ha, but we do have to place all of these things in the context that we are seeing changes, and there could be impacts of, for example, increasing temperature on increased OsHV-1 detection or emergence, etc. There are also other factors that can be related, including lowered pH or ocean acidification, precipitation, storms, and cyclones, though a lot of this is kind of in unknown territory. But what I often think about are organisms in the marine environment. It's an interaction of these stressors. It's also an interaction of all of these other human-caused issues that we have. And part of the reason I bring this up is that the next study, I'll talk a little bit about some of our temperature work and some of our ocean acidification work, though I'm not going to get into great detail about either of these factors. So I will move into talking about something completely different, though I think you'll see how it comes back to oysters. I tried to highlight the oyster side of our work here. But I did want to say that we've been working on seagrass wasting disease in primarily Washington State and the Salish Sea, really focused again on factors related to the host, such as demography, immune response to the pathogen, including diagnostics, looking at strain variation. We've conducted some ID50s, and finally, we're looking at virulence factors, as well as factors related to the environment, such as temperature and ocean acidification. So today, I will highlight another type of interaction. I'll highlight the interaction between oysters, eelgrass, and Labyrinthula. So what is seagrass wasting disease and what are the effects? So in the 1930s, seagrass wasting disease caused a loss of greater than 90% of seagrass beds on both coasts of the North Atlantic. And this did have huge impacts, including decline of waterfowl populations, lasting devastation to the bay scallop fishery, and extinction of the marine invertebrate snail. And this was, I know that in some areas that the eelgrasses have come back and are coming back, though in many areas they're not. And this is also a loss in key players for sediment stabilization and carbon sequestration. So what is seagrass wasting disease? I do want to pause and say this is one of my favorite sites in the San Juan Islands. It's a really beautiful area if you ever get a chance to go there. The hosts are seagrasses, clearly. The pathogen is Labyrinthula zoster, which I may refer to as Labby or LZ. And within seagrass tissues, it is these spindle-shaped shells shown here. We also often see necrosis and changes to the chloroplasts, which may be what that blackening we're seeing is. And this is kind of what these black, dark necrotic lesions look like. In extreme cases, you can have plant mortality, such as what happened in the 1930s. So what are the Labyrinthulomycetes? Well, Sabrina told us much better earlier this week, but they are unicellular marine protists. They're often known as marine slime nets containing an ectoplasmic net. This is our Labyrinthula on the top, the ostrochytrids, and then the aplanochytrids. And they're common in the marine environment, and they have a wide range of hosts, relationships with hosts. And in that, I mean, they can have a range from being commensal to pathogenic. And the pathogens are considered to be opportunists. And again, this Labyrinthula zoster is an example. Sabrina's favorite, QPX, is also an example. And there's an Aplanochytrium species that I worked with in sea fan corals that could be pathogenic. Working with sea fan corals and doing infection trials is nearly impossible, in part because most of the sea fans were already infected with this guy, which is probably no surprise. So what is the distribution of seagrass wasting disease? It's actually not just contained on the coast of the Atlantic and not just contained in Zostra marina, which it's primarily known as, and you might have previously heard of it as eelgrass wasting disease. We study it in the Salish Sea or the northern part of Puget Sound in Washington State. This is the San Juan Islands, and actually some of this is coastal Washington, not coastal, but inland Washington waters. And the light purple are healthy and the dark purple are diseased. So this is just to give you an example of the wide range of prevalence seen in some of our surveys. Of course, seagrasses are affected by lots of other issues globally, with estimates of losses of up to 8% per year. They're also affected by eutrophication, increases in temperature, natural disturbances, and it begs the question of what can we do? So one mitigation strategy are my favorite animals, the oysters, and I'm borrowing this from a previous publication, and I'll discuss the results of this. And we were really looking at a couple of components, whether oysters could filter the Labyrinthula and improve water quality, which would mitigate seagrass wasting disease, and while also whether seagrass could buffer pH and mitigate the effects of increased CO2. And of course, this is on top of the fact that there's a lot of concern, I think, on both coasts of the US that oyster aquaculture can negatively affect seagrass beds. So with the interest here of showing whether there might be a positive interaction with oysters and with seagrass. So of course, there are different types of filtration outcomes. We may or may not see,
Here's what I found.
Whoa, sorry. We may or may not see filtration of the pathogens. So in this first panel, the blue just depicts this as happy blue water. There are no pathogens present. In this depiction, let's say that oysters are filtering, the red is the pathogen, maybe that's Labyrinthula zostra. In this depiction, maybe it's oysters with OsHV-1 that are spreading it into the environment. And then lastly, they may not actually filter out the pathogen. So with this in mind, our primarily objective for a couple of experiments that I'm going to share was to examine the role of environmental factors on Labyrinthula zostra transmission and oyster filtration through co-culture. And these trials were all conducted at the University of Washington Friday Harbor Laboratories in the Ocean Acidification Environmental Lab. And so this was actually kind of the home base also for those surveys in the San Juan Islands. And in a first trial, which I would call, it was originally a pilot study to look at this. It was some funding that Maya Groner received and I was a collaborator on, was to look at whether oysters reduce wasting disease by filtering LZ, to see whether eelgrass improve oyster health by creating a local pH refuge, and also to see whether ocean acidification modulates wasting disease. And I should mention, if you're not familiar, in this kind of time frame, there were lots of issues and questions regarding low pH waters and ocean acidification in the Pacific Northwest. So this is really timely in looking at this sort of study. And what we did, we had 3 biotic treatments. So we had juvenile oysters co-cultured with eelgrass, and then we had eelgrass. And this is a phase 1. And then we had 3 PCO2 treatments, an ambient, so this was inflow, which was 656, a medium, a bad day, maybe in the Pacific Northwest, and then a high future PCO2. And we ran this first part of the trial for 3.5 weeks at a water temperature of 12 C, which you're probably thinking freezing cold. It was pretty cold. We did find that high PCO2, not surprisingly, reduced oyster mass. So in blue are, again, low, medium, and high PCO2. And there was no effect of the eelgrass, though we did see it modulated the PCO2 in the treatments, and I don't have time to really go into that data. I don't think it modulated it enough. We did, interestingly, in this first round of the trial, the plants from the field were naturally infected with seagrass wasting disease. And in this bottom graph, we did see in disease severity that oysters significantly reduced seagrass wasting disease, and that high PCO2, at high PCO2, seagrass wasting disease was lower. Interestingly, leaf elongation was also significantly greater at the middle PCO2, and seagrasses are one of these organisms that are expected to perhaps do well under OA conditions, so that wasn't a huge surprise. We took a subset of these plants and oysters and did a second round with just 2 biotic treatments, the combined and the eelgrass alone. And we had an ambient and a high PCO2. And the main reason that these values are a little bit different is the temperatures were increased. So they were at 15C and they were exposed to an infectious strain of LZ on day 24 of the original trial. And what we found was that unlike the naturally infected plants, we didn't see an effect of the presence of oysters in this experiment. The only, we saw an effect of our Labyrinthula zoster exposure. And this is both, again, severity, so this is the percent of the plant that's impacted, as well as qPCR, so the load per milligram of tissue. And we thought, this can't be right. So we went back in the lab, and my technician did a pilot study where these were with Prasostrea virginica, where she added actually a lower concentration of LZ into tanks with small beakers with oysters. And you probably already can tell that the oysters rapidly filtered out Labyrinthula zostra. And in fact, even this was literally right after she added, you can see the effect of the medium and large oysters. And it happened very early on. And so that was really exciting. And this was really the backbone of a new project that we wrote to Washington Sea Grant to further this work. Now, Natalie did also isolate LZ from the tissues of the oysters. So this will come up a little bit again later. So in one component of our Washington Sea Grant, we followed up on some of this work with Pacific oysters and their ability to mitigate seagrass wasting disease. We looked at whether Pacific oysters mitigate the disease, whether they can actually transmit it, and finally, how does temperature modulate this relationship? So this is an extremely elegant experiment that was run by my student Tori Agnew, where she had 5 experimental treatments, one with eelgrass exposed to Labyrinthula, so maybe you call this the positive control, the negative control, oysters with Labyrinthula, oysters and eelgrass, all 3 of them together, and then she exposed oysters to Labyrinthula alone and then added it into an eelgrass tank. And these were intended to be at 2 temperatures in triplicate. Of course, there was an issue that we lost one of the replicates, but then she looked at LZ exposure, oyster presence, and temperature and interactions on this. So first of all, oysters can decrease the severity of seagrass wasting disease. And I should mention she used a lower concentration of LZ. And we've also subsequently done work with ID50s and determined the ID50 is quite low, so it doesn't really take very many cells to transmit. But what she found, these are the control and the exposed, and she put little oysters where the oysters were present, were that as expected in the severity, labby exposure significantly increased severity, and also oysters significantly decreased severity. So oysters can decrease seagrass wasting disease in this fashion. Secondly, using qPCR, she also saw within the tissues the presence of oysters significantly decrease labby infection intensity. And I know that this seems small, but this is a log scale, and this data is in fact significant. She also, so I didn't show temperature previously. There was no interaction with temperature. However, temperature also impacted the relationship. So severity was significantly higher, as well as infection intensity at the higher temperatures as expected. She also found in that 5th treatment, based on the intensity, that oysters can transmit Labby back to the eelgrass. So this is based on that qPCR. Now, I think there are lots of questions here about the mechanism. We're actually going to follow up on this with some new funding to look at different densities of oysters, also look at, we can get into mechanistically what's happening. So she just plopped the oysters over. We don't know if it was on the outside or whether they were truly in the tissues. But excitingly, oysters can reduce both pre-existing disease and filter LZ. Oysters can also transmit LZ and seagrass wasting disease. And again, this mechanism is currently unknown. And there are some relationships with this disease and different environmental factors. So again, the disease was reduced at high PCO2 and increased at higher temperatures. So with that, I will make my acknowledgements quite quick, primarily acknowledging lab group at IMES, the Aquatic Animal Health Lab, as well as all of the folks that have worked on our OSHV-1 work, especially Karen Friedman and Kim Reese, as well as what I'm calling Team Labby, a lot of familiar partners, including the Friedman Lab, but also Drew Harvell and Maya Groner. And a special thanks to all of the oyster farms that either collaborated or provided oysters. So I am more than happy to answer any questions. I'm sorry if I talked too long. I just couldn't delete anything.
Okay, we have one in the chat. Let's see.
Hi, this is for me. It's
Yeah,
hi. I have to go to another meeting in like about 1 minute. So I put it in the chat. Hi, Colleen. Thank you. It was a very interesting talk. Whoops, I can start my video. Colleen, it's very interesting.
Hi, Diana.
But so it was really interesting, and all your work was actually, this was really pretty exciting stuff. But you know that Cress ostragigus is a non-native species, and it's very invasive everywhere that it's being grown for aquaculture around the world.
Right.
And it's been shown around the world to have serious negative impacts when it escapes aquaculture because it is such an important invader. And in the San Juans where you were working, the only time you're going to see gigas with Zostra is going to be if it's planted there for aquaculture. So given that, what would you recommend for potential use of these oysters?
The reason we use the San Juans is to use the facilities there, as you know, which are quite good, and the eelgrass beds are quite nice there. But as you're aware, the rest of the industry in Washington also grows oysters, right? And you know, all of the Pacific coast and most of the East Coast grow oysters, right, in areas where there are eelgrass beds.
Right.
So that is the primary, you know, reason for conducting research there. And yeah, I know that gigas is considered invasive, but I think the realistic part of this is that it is an aquaculture species that is here to stay. I think it would be really interesting to look at other species like Ostreid, you know, the native oyster, or other specific native species as well. But in, you know, this sense, this funding was to kind of look at, you know, both kind of from the disease ecology standpoint, as well as from an eelgrass protection, as well as from the aquaculture side. So I see what you're saying, though I think that gigas is there to stay, and I understand your concern as well.
Well, it's only there to stay in aquaculture where it's currently grown, which is mainly down sound.
Mm-hmm.
But deliberately outplanting it in places like the San Juans will enhance its likelihood, right? So there's,
Oh, of course. And that's not what we were
it's
suggesting
just
there.
care in the messaging.
Yeah, yeah, of course. And in most senses, this messaging is not for growing it in places like the San Juans where there's
Right.
not a lot of oyster culture, but it's for more broadly the industry.
Right. But very interesting. Thank you very much.
Thanks for the question.
Anybody else have any questions? You can either put it in the chat, raise your hand, whatever works.
I was either really clear or talked way too fast. I'm not quite sure which or both.
Hi, Colleen. I was going to save most of my questions for talking to you later, but I am curious what you think is going on with the herpesvirus variants. Are they sort of already all over the place, or are there new ones evolving, or they're coming from a reservoir or some other organism? Or, I mean, what do you think is happening?
I don't know. So it seems in San Diego that it could have come from a live market, this is a hypothesis, or it could have come from ballast water. So I haven't been to the Flupsy for lots of reasons, but I did see there are a lot of live markets on the waterfront. And in talking with folks in Australia, some of their first outbreaks occurred in areas near live markets. They were also big shipping harbors. And I'm sorry, I forgot to mention the shipping harbors. I don't know about it being everywhere. I've often argued this point, especially with the Europeans, because I think we have a pretty good sense of where it's not in the US, at least from the Pacific oyster perspective, because Ralph Elston does a lot of testing. I've done, testing of all of our pre-samples. Our Sentinel program really nicely showed, I didn't show all of the data, that, Washington and Oregon were negative, while, the 2 sites in California that we expected were positive. So yeah, I mean, that's the $1 million question. Where did this come from? I suspect it came somewhere from Asia, came over somehow, but I have no idea.
Okay. At least that's the right question to ask. Oh.
I agree.
Yeah, cool.
To jump over Jackie's question, Colleen, the fact that that microvar disappeared in San Diego for a couple of years, right? A year and a half? How long it did disappear before popping up again? Is it actually what popped up? We don't know yet if it's the microvar or not, correct? Yeah.
It's the same variant.
Same variant. Yeah, okay.
Based on sequencing of that specific genome region, it was identical. So, and it wasn't just my sequencing, but it was also the US National Reference Lab. So it seemed.
Yeah, go ahead, sorry. No, so it did disappear and show up. And then the seed.
That's not uncommon.
Yeah. You mentioned the seed was tested, Colleen. You mentioned
the seed was tested before it went out.
It was negative. Yeah. Okay.
Yeah. And none of the seed that has ever gone in there is tested positive. So and the seed that has gone in from multiple locations. So the first seed that went in came from a different bay than the second one. So it seems pretty clear that it's in San Diego and not other locations. And in GenBank, this was a new variant. There wasn't anything specifically identical. I mean, we'll know more when the full genome sequencing is done.
I also had a question about the oyster herpesvirus. Is my sound coming in good? Because sometimes it comes in weird off my mic. Okay, good. Do you think there's any possibility that it could be transmitted through shell? I know it's not hard to find oysters from all over congregating in any type of city. I know you can buy oysters from all over the world in New York City, for example, and people don't tend to care about their shell too much. And I don't know how durable herpes viruses are, but if it's a low LD50 to get in there, one of those, like one case can rapidly explode.
I don't think that anyone actually knows the answer to that question. My feeling is it wouldn't remain on the specifically on the hard surface of the shell for very long, though maybe it could be associated with communities, or if there are still tissue on the shell though, say if there's a bit of gills or mantle on there, that's where I would be concerned. I've been asked that many times, and no one has really looked that closely and has a good answer for it. But no, that is concerning. And I know that Bassem knows about this. There's been a lot of talk of reopening trade with Europe and bringing in animals from infected parts of Europe, so which was halted. But yeah, that's definitely a good question. And the farmers I work with in Tomales Bay, someone one day brought in oysters from New Zealand to their farm to share them with them, and they were just aghast. And they made sure, not to put them anywhere near the water. And it was just kind of this horrified sort of sense.
Thank you.
You're welcome.
Colleen, obviously the results of the comparison of the strain is very interesting, extremely interesting, intriguing, and clearly deserve further investigation about understanding. But you mentioned that some of these strains may come from a line that are adapted to different salinity. Is this correct? Some of what you tested in that lab experiment with the OSHV?
Oh, so the Virginica, yes, not the Gigas, but the Virginica were like the low and high salinity Virginica. And in fact, we had to run the trials at different salinity because we had a really hard time acclimating the oysters.
Okay. This was my question indeed.
Yeah, so we did the Gigas at more standard 32-ish, and I believe the Virginica at 20. We weren't able to get the West Coast Virginica that we could have held at the higher salinities. Yeah, it just was one of those things. And of course, salinity could impact this whole relationship too. So, but there was really no way around it, at least in that trial.
And temperature, salinity, obviously the most obvious one, but yeah, you need to run the test at given standards.
Yeah, so the temperature was at about 22. That's very standard for OsHV-1 testing. Of course, it may be variable across that temperature range too as well. So yeah, and that's a huge question. I think that's a huge question for Gigas and Virginica cultures, really those questions of salinity and temperature and how the response may be across the environment.
Interactions.
Yep, exactly.
In some of the challenge experiment, there were some drop between day 4, I believe, and day 6 in viral loads. Is it? Yeah.
It wasn't significant, but there was. It looked like a drop, and it may just be there's a lot of variability in the data. Wasn't a lot of data collected. We've been collecting more animals and also endpoints since that time. So, we would expect it to drop off, especially if these are, hopefully survivors. But yeah, in that data, there wasn't anything statistically significant.
Yeah, I'm saving some of the questions for the discussion later as well, but I know that the shooting.
Great. All right. Thank you, Colleen, for a wonderful seminar.